Now we'll consider mutations, or changes in the genetic code. Often permanent changes do occur in the genetic code. And it is these permanent changes that are the driving force of evolution. Mutations can involve any change to the nucleotide sequence, or on a much larger scale. This is a loss of base pairs, addition of base pairs, or rearrangement of a whole order of a segment of DNA. Some of these mutations result in positive changes, some have a negative impact, and some have no impact. You might recall that there's some wobble in the third nucleotide of each triplet sequence, such that the same amino acid is specified, so essentially we end up with the same protein. So we define a mutation as any change in nucleotide sequence. The wild type of an organism is the microorganism that exhibits the natural or non-mutated characteristic. And you might say, well, with so much change going on in microbes, how do we know what's the non-mutated characteristic? We tend to say that it is the most common phenotype. The mutant strain is when a microorganism bears a mutation or is not as common. These mutant strains contain variants in morphology or nutritional characteristics or their resistance to certain chemicals. They might even change temperature preference, and nearly any enzymatic function can be altered. These mutant strains can be useful for tracking genetic events, unraveling genetic organization, and pinpointing some genetic markers. We can test for mutations using this technique of replica plating. In replica plating, we're going to culture a bacteria, and it has discrete colonies. And then after that plate is exposed to a mutagenic agent, say some radiation, we'll take a sample off of that plate. There's a special velveteen substance on this carrier. When you press it onto the plate, it picks up cultures. We can stamp some culture on this plate, where we have a medium that selects for that mutant, and then we have another stamp on some non-selective medium. So anything can grow here. So basically over here, only things that possess a certain mutation can grow on this culture, and here anything can grow. So now we've isolated colonies of mutant strains. What causes mutations, you might ask? There can be spontaneous mutations or induced mutations. Spontaneous mutations result from random changes in the DNA that arise from errors in replication. We've already explored these, and we know there's plenty of machinery to fix many of these mutations, but not all of them are caught. Induced mutations result from exposure to known mutagens. In this table over here on the right, we have a sample of known mutagens. There are chemical mutagens that have effects on specific base pairs. And there are radiation mutagens, like ionizing radiation, gamma rays and X-rays, as well as ultraviolet. These can introduce major physical changes into the DNA, and that's precisely why radiation is a very effective microbial control agent. Many of these chemical mutagens also act as carcinogens when exposed to vertebrates. So if we're exposed to them, they could cause cancer for us. It's only because of the rapid rate of bacterial reproduction that we can actually observe mutations at this high rate that we do in bacteria. It's much more easy to see what's going on in prokaryotes than it is eukaryotes. Mutations can range from very large mutations in which there are huge sequences that are gained or lost in a chromosome, to very small ones that only affect a single base. These mutations are broken into categories. There are either point mutations or frameshift mutations. Point mutations involve addition, deletion, or substitution of single bases, and you can end up with a range of different results. The missense mutation is where we have any change in the code that leads to a placement of a different amino acid. You can see an example of a missense mutation here. If we're changing just the letter T in this codon here, so that there's a T instead of a G that we see up here, then we end up with an RNA that has GAA instead of GAC, which means that we'll substitute the base into glutamine instead of aspartame. Now, this change of amino acid could have a range of effects. It could make the protein non-functional. It might have no effect at all. or it could change the function of the protein. Perhaps it operates better at a different temperature. Now let's look at a nonsense mutation. This is where the changes cause a normal codon to become a stop codon. Then a silent mutation might offer a base change where the original G is turned into an A, and that results in a different triplicate code on the RNA. However, it does not change the amino acid in the chain, so this is a silent mutation. Occasionally, we'll see that a mutation occurs, and then that same base is mutated back to its original state. This is called a back mutation. So the gene has undergone a mutation that reverses its original base composition. Sometimes a mutation of one nucleotide, like through an addition or deletion, could cause a whole frame shift. For example, here, we've literally pulled out a G, which means that the whole thing is now off frame. So this C is now going over here, and each thing downstream of that is now a changed triplicate. This frame shift could cause a number of different effects. It could lead to a premature stop codon that you see in this case, or it could simply lead to a protein with many changed amino acids. In this case, you probably have a non-functional protein, so frameshift mutations really have the largest effect. Now earlier we learned that DNA had its own proofreading mechanisms to repair mistakes in replication. The cell also has additional systems for finding and repairing DNA that's been damaged by these various possible mutagenic agents. Most ordinarily, the DNA damage is resolved by some enzymatic systems that are specialized for finding and fixing such defects. There are a couple of examples. For DNA that's been damaged by UV radiation, it can be restored by photoactivation or light repair. This is mediated by the enzyme DNA photolyase. It's a light-sensitive enzyme, and it detects damage in the DNA, attaches to that site, and repairs it. Another common mechanism is excision repair. In excision repair, we see that a couple of enzyme complexes come along. First, we have one enzyme complex that comes along and it cuts out the bad base pairs. And then a second enzyme will come along and bring in two new base pairs, the correct ones. And it really does this by reading what's on the complementary strand. And then it glues those into place, so now we have the correct base pairs in place. Let's take a look at a quick video of how this process works.
UV light and other ionizing irradiations damage DNA molecules by creating thymine dimers between adjacent thymines in the DNA chain. The dimers weaken hydrogen bonding and lead to deformations of the double helix that create subsequent errors in replication resulting in mutations. To protect cells against such damage, DNA photolyase, a light-dependent DNA repair enzyme, eliminates thymine dimers from the DNA strand. The enzyme binds to a thymine dimer, and upon photoactivation, transfers an electron to the dimer, leading to the reformation of two thymines. The electron is subsequently returned to the enzyme after the dimer has been eliminated. Chemical agents can induce the methylation or alkylation of guanine residues in DNA at the oxygen attached to the 6th position of the purine ring. These methylation changes can result in mutations. The enzyme methylguanine DNA methyltransferase eliminates these methylation events and restores the correct structure to guanine in DNA, preventing mutations from occurring.
Now, with the advent of new agricultural, industrial, and medicinal chemicals that are constantly being added to the environment, the exposure to them is widespread. The discovery that many such compounds are mutagenic and that many of these mutagens are linked to cancer is significant, as I'm sure you're probably all aware. Although animal testing has been a standard method for detecting chemicals with carcinogenic potential, a more rapid screening system called the Ames Test is also commonly used. The experimental subjects in this test are bacteria. The premise is that any chemical that's capable of mutating bacterial DNA can similarly mutate mammalian and thus human DNA and is therefore potentially hazardous. One of the indicator organisms that we use in the Ames test is Salmonella typhimurium, and it's lost the ability to synthesize the amino acid histidine, so it has to have histidine provided for it in order to survive. This defect is highly susceptible to back mutation. That is, the histidine negative can become histidine positive. It can synthesize its own histidine, and thus it can survive on non-supplemented media. Let's take a closer look. Here we have our tube of Salmonella histidine minus, meaning it can't synthesize its own histidine. In the control setup over here on the left side, the bacteria are going to be plated on a histidine-free medium. So there is no histidine on this medium. And so the bacteria cannot grow unless it has a back mutation to be able to produce its own histidine. The experimental plate is prepared in exactly the same way on a histidine-free minimal medium, except that this plate contains the test agent, the thing we're wondering whether it has a carcinogenic effect. So these plates are incubated for about 12 hours. Any colonies that form on this plate and any colonies that form have to have back mutated to become histidine competent. That is, they can make their own histidine. Otherwise, they simply wouldn't be able to survive. Now, there's a background rate of back mutation in which even in the control plate, we would see some hist-positive colonies arising, growing on this plate. However, if there's a significant difference between the number of colonies growing on the control plate than there is the number of colonies that are present in the plate that contains the chemical, then we know that the rate of back mutation is much higher on this plate, and thus the test chemical must be an issue. A pretty simple dynamic test. Many mutations are not repaired by the cell's mechanisms for repair, and how they're managed really depends on the nature of the mutation. Most mutations are permanent and inheritable, and most of them are harmful, which cause the bacteria to die. However, some do provide adaptive advantages. If the mutation codes for a non-functional protein and there's only one copy of that protein, that is, the organism is haploid, then the cell probably won't survive because without a functional protein, it cannot do at least one of its necessary processes. However, a small number of mutations actually end up conferring an advantage, which increases the survival and reproduction of an organism. That survival could be increased in the present time, or perhaps in the future when the environment changes. For example, if it suddenly got more cold, or much more warm. In this sense, the preferential survival means that offspring will reproduce more frequently, and thus the mutation will be passed to the offspring and remain in the population. This is simply how the process of evolution works, and bacteria do it at a very high rate. So let's have a quick concept check. What type of mutation is this, where we see that the original triplet said ATG encoded for methionine, and now it's become ATC, so the third place in the triplet code has been changed. We've replaced the amino acid. It's not silent because the amino acid was replaced. It's not nonsense because we still have some sense out of it. Amino acid is still being coded for. But it is missense because it's not the right amino acid. And this change could cause a big change in the protein structure. Maybe one was very polar and now one is nonpolar, thus changing that whole region of the protein and its interaction with other portions of the polypeptide. Or it could be the same kind of polarity, which would result in very little change, and probably the enzyme would still work. So not all mutations are bad, a few of them are good, and sometimes they can confer a preferential survival and reproduction, and thus we have evolution.